So serine threonine specific protein kinases are important group of protein kinases that forms the basic entity of signal transduction. So basic proteins which are responsible for phosphorylation, and phosphorylation is an important means of signal transduction pathways. So that is by the kinases, protein kinases, and serine threonine specific protein kinases are the particularly important because those are the kinases which are versatile in function. Most of the signal transduction pathways are mediated by serine threonine and also tyrosine. So serine threonine, why? Because they are the protein kinases which were first discovered. So afterwards, tyrosine specific protein kinases. Already we discussed the classification and what actually serine threonine specific protein kinase. These all things in the last classes we have discussed. And today, the after the basic discussion of serine threonine specific protein kinases, in the next few classes we are going to discuss with reference to few examples of these serine threonine specific protein kinases, and those are very important for your big answer questions, and as far as the conceptual understanding also in the signal transduction, these kinases are very important. So first one is protein kinase A. So today we are going to discuss about the structure of protein kinase A, then its subdomain structure, then how it is being controlled. Control, you should understand now by this time, many times I had repeated in the class, control or regulation means plus or minus. So the activity either induced or suppressed by what mechanisms? That is what control, and that we are going to discuss today about the control of protein kinase. So protein kinase A, basically, this is also known as CAMP dependent protein kinase. So this is basically a member of superfamily known as AGC kinase. Protein kinase A is very sensitive, that depends on for its activity cellular CAMP. CAMP is an important second messenger. If you recollect, we discussed in second messengers. So CAMP is an important second messenger, and depending on the level of CAMP, cellular CAMP, important kinase, that is protein kinase A, activity is dependent on. So this protein kinase A is one of the kinase belonging to AGC kinase. What is AGC? Protein kinase A, G, and C family. So here you see. So this includes, along with protein kinase A, protein kinase B is also known as AKT, PKC, and PKG. G here is CGMP dependent. Like PKA depends on CAMP, and PKG means CGMP, another second messenger. So that influence the activity of PKG. That's why it is called as PKG or CGMP dependent kinase. That's why these all together constitute AGC kinase superfamily. So PKA belongs to superfamily of AGC kinases. So amongst all these different kinases of AGC superfamily, protein kinase A is well known and well characterized. So the cellular CAMP, as we discussed, that is the important one, which responsible for the regulation of activity of protein kinase A. So CAMP is the second messenger that influence this protein kinase A. That's why it is called as CAMP dependent protein kinase, nothing but protein kinase A. So in the absence of CAMP, what happens? Protein kinase A will be in inactive state. When it is inactive or inhibited, it is existing in tetrameric state. See here, R2C2 bound state. Again, if you remember this protein kinase A, in analytical biochemistry, we had discussed. So that was regarding FRET assay. So FRET assay mainly fluorescence resonance, energy transfer. So when two subunits or two fluorophores are present within the distance of 10 angstroms, then if one fluorophore is excited, there is a non-resonating energy transfer to other subunit taking place through FRET. That is FRET energy transfer to other fluorophore. So instead of A fluorophore, which you have excited, you will see characteristic color of the B fluorophore. Understood? So you have A like red, B as yellow. You have not excited the B fluorophore, which emits yellow color. Rather, A fluorophore, red color one, you have excited, but you are not seeing red color. Instead, you are seeing yellow color. This is because of the fret. So the concept mainly relies on the distance between two of these fluorophores. So in that concept, we discussed as an example R2C2. So when these regulatory and catalytic subunits are together in the hologen chain, so two regulatory and two catalytic subunits, they are in the inhibited state, right? So when they are going apart means it is getting active. So activated state means fret will be lost, and that depends on CAMP level. Do you remember all of you this example we had discussed, correct? So if you don't remember, again go through that fret and example, you can make out that R2C2 in its bound form, when there is a fret signal, it is inhibited, it's in inactive state. So that's what here we are discussing. So in the absence of CMP, it exists in a tetrameric state of R2C2 domains. So now pKa has several important functions in the cells and mainly in the glycogen, sugar and lipid metabolism. Then two scientists, Fisher and Krebs. These two scientists, they won Nobel Prize in 1992 for elucidating the phosphorylation and dephosphorylation mechanisms by protein kinase A. So protein kinase A work has given one Nobel Prize in biochemistry. So protein kinase of AGC family, they have the same structure. All these protein kinases of AGC, that is protein kinase A, pKa, pKb, pKc, and pKg. All these protein kinases have the similar catalytic domain and are regulated by similar fashion of phosphorylation in the activation loop. So mechanism of phosphorylation is same and the structure of catalytic domain of all these AGC family of protein kinases is same. So inactive, as we discussed, it is nothing but the total tetrameric state of the R2C2. Okay, and when it is activated, so activation mainly because of the CAMP levels, which are responsible for binding to R2. So you see, there are four CAMPs which are binding to two of the R subunits and plus 2C. So CAMP bound state of this protein kinase A is activation. And there are different isomers, mainly regulatory subunit if you see 1 and 2. And it 1 and 2, again 4, each one has alpha beta. So R1 alpha beta, R2 alpha beta. So 4 subunits are the isoforms of regulatory subunit, and as well, catalytic subunit exists as alpha, beta, and gamma subunits. Okay. So activity of protein kinase A is mainly controlled as we discussed this. CAMP is the messenger that influences the activity. In the absence of CAMP, PK is existing as a tetramer in a bound form of two regulatory subunits, each containing two CAMP binding sites and two C subunits. So regulatory subunit has got binding sites for CAMP along with the catalytic C2 subunits. The catalytic activity, so catalytic activity, that is the function of protein kinase A, active site is present in the C subunit. That's why it is called catalytic subunit. It is having the catalytic domain, active site, and that is masked when CMP is not there, when it is in inactive state. that is masked in the hollow enzyme by the C2R2 as the R subunit. So in the R subunit, regulatory subunit has got, already we discussed, one binding site for CAMP. There are two binding sites R is having, like that R1, R2. So totally four CAMPs are binding to two of the R2 subunits of the protein kinase A, and another subunit, that is inhibitory domain, also present in the regulatory subunit. So this inhibitory domain is the one which is responsible for masking this active site. Where is the active site? In the C subunit. So when they are in bound form, R2C2 in inhibitory state, the inhibitory domain of the R subunit is masking the active site of the catalytic subunit. That's how pKa is in inhibited state, inactive state. Understood, all of you? So now we discussed this protein kinase is existing in different isoforms of multiple R and C subunits. Okay, and each of these subunit will have a specificity of its action and variability of its function. That is why pKa is involved in many more signaling pathways and cellular responses because of its different isoform structure. All of you understood what I said? So this is what we discussed. So this is inactive state, R2C2, so 2R and 2C. This is bound state, and in bound state, through this pKa is bound to, this is A kinase anchoring proteins. So there are A kinase anchoring proteins which are bound to this pKa, through which it responsible for the downstream signaling activation. So pKa is a protein kinase. When this kinase is getting active, it acts on another kinase by phosphorylation. So this protein kinase undergoes phosphorylation and getting activated. Then phosphorylated protein kinase A, which is active form, again can phosphorylate another kinase. That is what signal transduction. Correct. So here you see this is R subunit. There are two R subunit, two catalytic R2C2 in bound form, and each R has got two of the sites here mentioned. These are CAMP binding sites. So there are four CAMP binding sites on inactive protein kinase A, and this depends on the CAMP level in the cell. So this can undergo CAMP hydrolysis and it can form AMP. However, the CAMP, when it is present in the cell, responsible for activation. When it is binding to this R subunit, total four CAMPs responsible for activation. So activation we discussed in inactive form. The R is having another site which is not mentioned here, that is inhibitory site. This inhibitory site will mask the active site present in the catalytic subunit. That's how the phosphorylation of the catalytic subunit is inhibited. So for getting active, fully active form of protein kinase A, one requirement is binding of CAMP. Second requirement is the catalytic subunit as well R also, it has got phosphorylation sites. pKa should get phosphorylated. So phosphorylated CAMP bound form of protein kinase A is fully active form. Understood? So that's how now in the inactive form I said, due to inhibitory domain of regulatory site, that is masking the catalytic site of the C subunit, the activity is being inhibited in inactive form of pKa. Now, when CAMP is binding, so here first CAMP will bind, and this binding is cooperative binding. You know cooperative binding? Anybody? Can anybody tell the example? What do you mean by cooperative binding? You should know. You are in second year now. Can anybody tell what is cooperative binding in proteins? Enzymes particularly. Sanjana?
Sir, probably the oxygen and hemoglobin
Very
binding.
good, very good. So what is it? Tanisha, I didn't ask example. I just expected what is cooperative binding, but you are one step ahead. You give an example. So Tanisha, now others should get what is cooperative binding. Punyashri, Punyashri, can you unmute?
Yes, sir.
What is cooperative binding? Punyashri,
I don't know, sir.
you don't know. Okay. Pooja,
Yes, sir.
Pooja, what do you mean by cooperative binding?
By binding with the substrate, it will bring some changes, sir, in the
Yes,
substrate.
exactly. So those changes will activate further binding. Okay. What you said is half still. So when oxygen is one oxygen, so hemoglobin is, there are how many subunits? What type of molecule hemoglobin? Beta and alpha, two beta, two alpha. So four, right? So to one of the globin, when oxygen is binding, so binding of the substrate is responsible for change in the conformation of the molecule in such a way that the other oxygen molecules will bind with more felicity. Understood all of you? That is what cooperative binding. So same thing even in this case also you can see the cooperative binding. So what happens? There is CMP binding is taking place one of the site in inactive. Then what happens? This undergoes conformational change and it facilitates in such a way that it's responsible for binding of other CAMP in more with more affinity. So that is what cooperative binding. So this activated CAMP occupied active form of protein kinase A, then with the change in conformation, loses the affinity between the subunits. So they will get disassembled. This holoenzyme gets disassembled. So R subunits and catalytic subunits will come out of this R, and this is now completely active form, this catalytic subunit, which is responsible for activation of downstream protein kinase, downstream kinases. So catalytic subunit has got a larger lobe where active site is present that it can occupy the substrate and responsible for phosphorylation of the substrate. This is happening not only with occupying occupancy of the CAMP here, but also this undergoes phosphorylation, right? I said pKa, CAMP bound, and phosphorylated. It undergoes autophosphorylation, and that is fully active form. So any questions you have? All of you understood now about the structure of protein kinase? These are very important. If you follow, it is very easy. Alodi, Alodi, have you followed?
Yes, sir. Yes, sir. I have followed.
Okay, good. So CAMP, as we discussed, the binding is cooperative manner. So two sides termed as A and B are present on R subunit, right? So two subunits are having A and B. Total four binding sites are there, A and B. In the inactive form of coenzyme, as I said, the B site, okay, is exposed. One of the site in the inactive form is exposed and it is available for binding of CAMP. So when it is inhibited, the B site of the R subunit is being occupied by the CAMP. However, when B site is occupied, this enhances the binding of CAMP to the A site in a cooperative fashion. Understood. So B site is exposed for CAMP binding in its inactive state, and when it is bound to CAMP, it is responsible for cooperative binding of other CAMPs at remaining A sites. So the binding of CAMP to the R subunits leads to large conformational change in R, okay, as revealed by the crystal structure of C subunit in complex with fragments of R1. So this is what cooperative binding with the binding of CAMP induces the conformational change. So in the absence of CAMP, what happens as we discussed, R1, the regulatory subunit, wrapping around the larger lobe of C subunit, the larger lobe catalytic active site that is being wrapped or masked, making what happens, how it protects in that way. When it masks that active site in the catalytic subunit, the intense contacts with activation loop. So this inhibitory domain of R is making intense contacts with the activation loop and covers, basically it is covering in that way substrate binding site. So substrate binding site is present in the C subunit, and that active site or substrate binding site is being masked by the regulatory R1, which wraps around this particular larger loop domain of the C subunit. Understood? So this way when CMP is absent. So the binding of now 4 CAMP molecules, 2 to each R subunits, that leads to cross conformational change in the R. So when they bind, it responsible for change in conformation. So the inhibitory domain unmasks the activation loop. As a consequence, affinity between the R and C is reduced. So within a particular affinity, this R2C2 tetramer will be there, and that affinity will be lost with the change in the conformation. So holoengine then dissociates into dimer of the CAMP bound R subunits and 2 monomers of C. So C are getting dissociated. So it remains a dimer of 2R and the remaining 2C. So how many you are getting? 3 subunits. 2R together, dimer. This is monomer. 2 catalytic subunits are getting disassembled into individual catalytic subunits. Okay. So that way it is getting activated. As I said, the activation is not only by CAMP binding, also it requires to undergo phosphorylation. So this is the domain structure. If you get subdomain structure of the pKa, you have to write like this. So full catalytic activity of C subunit requires phosphorylation. It has to undergo phosphorylation of 309 residue, and that is particularly 309 197. This is the site present in the activation loop, and this is present in the C subunit, right? Activation loop. So this loop functions as major binding surface for the R subunit. So this is the site which is being masked by the R subunit. So catalytic subunit with the catalytic loop containing 309 residues have to undergo phosphorylation. Then it's becoming activation. So there is a conserved sequence within the C subunit that undergoes phosphorylation of protein kinase A. So you see here, these are, you have to remember like this: 2-arginine, X, serine 309. Okay. Now, variants, the other sequences, lysine, either at one place or the first place or two places. Okay. So these are the conserved sequences that undergoes phosphorylation, particularly at 309 197 residue. Understood? So the R2 subunit contain such sequence and is therefore subject to phosphorylation by C subunit in the holoengine without the release of inhibition. So phosphorylation, even R2 is having, and catalytic is the one which is responsible for autophosphorylation of protein kinase A. And then R1 subunit is having the binding sites for ATP and Mg2 plus. So these two form the stable holoengine complex, whereas R2 subunit do not form. So here R1 has got ATP Mg2 plus binding site, R2 subdomain has got phosphorylation sites, and C subunit has myristic acid residues which is of unknown function at the N terminus, along with serine threonine phosphorylation sites. So both C subunit and R2 subunit have got the sites for phosphorylation, and C subunit has got the myristic acid residue along with the serine threonine sites for the phosphorylation. So this is the one which is having threonine 197, which undergoes the phosphorylation, and it is responsible for change in conformation and activation of this protein. So these are the domains. That's what we discussed. So we discussed that R regulatory subunit exists as 1 and 2 forms, and 4 are there of alpha and beta types. Then catalytic. So you see here, these two different forms are variable with respect to the subdomain structure and its function. There is an autoinhibition loop, as we discussed. The autoinhibition or domain inhibited state is because of the inhibitory domain masking the activation loop in the catalytic subunit. Okay, so that is the autoinhibition loop here, and you have CAMP binding site is there here. You have phosphorylation sites over here, right? So A and B sites are there in the regulatory site, and same way here for R2 beta. Then you have catalytic subunit. Here, as we discussed, there is Mg2 plus and ATP binding site is present, and then the larger lobe, which is a substrate binding active site, C lobe and N lobe. Understood the subdomain structure? So pKa. Next point is control. How the control of protein kinase A is being taking place in the cell. Control, as we discussed, regulation or control means induction of activity or repression of its activity. So protein kinase A, when it gets activated, it can activate a variety of the downstream signals, and this activation mainly associated is in the depiction you see. This is through the proteins known as A kinase anchor proteins, which are associated like here. So these proteins are associated with these catalytic subunits, the R subunit and C subunits of the protein kinase A, and these have got important role in activation. So upon activation of pKa, the C subunits are released, phosphorylate substrates that are located close to this A kinase anchor proteins. Okay. So this is the way how this AKAP proteins also again now responsible for the regulation, either to induce or to inhibit. Then substrate phosphorylation can be down-regulated by binding of pKa inhibitor. So negative regulation is because of the presence of pKa inhibitor proteins are present, and that can inhibit the activity of this protein kinase A. So negative feedback is because of the presence of enzyme known as phosphodiesterase. So pKa inhibitors are responsible for controlling phosphodiesterase. See here, this is what we discussed. So CAMP responsible for activation. Now this activated form of pKa is down-regulated. So this is inhibitory signals. Inhibition means suppression of the activity by the pKa inhibitor. What it causes, it suppresses the phosphorylation of the PD. PD means phosphodiesterase. This phosphodiesterase is responsible for the conversion of CAMP to AMP. When phosphorylated PDU is acting on the hydrolysis of CAMP, there is no CAMP, so activation is inhibited. That's how it is inhibition. So please, all of you note down now. Once we discussed, phosphorylation can be inhibited. Mostly phosphorylation is activation. But here you see in this example, phosphorylation is inhibitory. So phosphorylation of phosphodiesterase is inhibiting the activation of protein kinase A. Understood? So that's how now protein kinase A inhibitor is controlling the negative regulation of this protein kinase A. Understood all of you? So what is happening here when pKa inhibitor is inhibiting this step, then there will be no phosphorylation, then there will be more of CAMP. So this is the reverse of that. Phosphorylated PDE is responsible for negative regulation. When it is getting phosphorylated phosphodiesterase, active form is suppressing the activation of pKa. Understood, all of you? Any questions you have? Sangeeta?
No, sir.
So all these illustrations, you have to make notes for your answers. You can write here one more site here, okay? That is inhibitory site. This are also having. So if there are no questions, that is all for today's discussion. All of you please go through. I'm not sure how much you have followed because this is all in-depth discussion about this serine 309 protein kinases with respect to protein kinase A. Unless you read again and again, make notes, and you may feel now, yeah, it is very easy when I explain. But for you, you have to again reproduce this because in the exam you have to write the answers, right? So please write today itself notes for all these things. All this material is from your Gerhard Krauss book you have, and you can directly straight away make the notes for these topics. Protein kinase structure is important, and then its regulation. So no questions. All of you understood that? Soumya, have you followed? Please unmute and tell me. Soumya, are you listening or not? Is my voice is audible?
Yes, sir.
Soumya, are you there? Vidya,
Yes, sir.
have you understood Vidya?
Yes, sir. I have followed.
Okay. Okay then. So I will stop here and tomorrow we'll continue the discussion. But 19th, as I said, your offline classes supposed to start.